Welcome back, everyone. Today we're going to be talking about genetics part 1. So with genetics, we'll talk about Punnett squares, and it all goes back to the father of genetics, Gregor Mendel, where in the 1800s he started studying pea plants and trying to see what actually determines or how are information transmitted from one organism to the next in reproduction. So he was trying to study how this hereditary material might be passed down. So based on what the parents look like, what do the offspring look like? And he noticed a lot of interesting trends. If he cross-pollinated certain pea plants, then the offspring would, maybe it was round and wrinkled seeds. And for example, perhaps all the offspring had round seeds. And then he would be like, okay, that's strange. And then he'd cross-pollinate those, and maybe 3/4 of the offspring of those plants would have round seeds. And the predictions were starting to match the observations. And he was able to come up with a lot of trends that he noticed in these pea plants. And he would cross pollinate thousands of them and record the properties of the offspring and what the parents looked like to try to see what kind of trends were there. So, genetics is the study of heredity, and we call Gregor Mendel the father of genetics because he was really the first person to quantify some of these genetic observations. So some general terminology, we'll use chromatin, chromosomes. Chromatin is when it's uncoiled. Chromosomes is when the DNA is condensed. Usually see that with reproduction. We'll talk about genes. A gene is just a sequence of DNA that codes for a trait, a unit of heredity. So an example might be eye color. You have DNA sequences that code for your eye color. And then an allele is a possible form of a gene. So an individual will receive 2 alleles for each gene. So an allele, we might represent an allele with just one letter. That'd be one allele. But your genotype, because you receive a chromosome from your mom, a chromosome from your dad, you have 23 pairs. So you might have 2 chromosomes, and one could have been a dominant allele you received from your father, then maybe a recessive allele for eye color you received from your mother. So if we say a locus, that's the location of a gene on a chromosome. If it's homozygous, that means it's the same. That would be homozygous dominant because they're both capital letters. We use capital letters to represent dominant alleles, which are those that when they're present, they are expressed if they're dominant. Heterozygous, hetero means different, homo means same. So heterozygous would be a difference in the zygote or different alleles. So in this case, dominant and recessive. Or we could be homozygous recessive. Homo meaning same, and then zygote, the same genes or the same alleles in the zygote, which would be what you get at fertilization. So in this case, it's going to be the same, but they're going to be recessive, so we would call that homozygous recessive, and then different is heterozygous. Genotype is when we're talking about the letters. So that might be someone's genotype. That could be someone's genotype. Maybe we're talking about another trait. That could be a genotype. So genotype is the set of alleles that someone has, and we represent that by letters. Whereas a phenotype is the physical appearance. So this is someone's genotype, capital B, lowercase b. The phenotype might be brown eyes. Think physical. Phenotype means what it physically looks like. Wild type is going to be that most common phenotype or allele for a gene in a population. You'll see us reference that when we talk about fruit flies a lot. For whatever reason, that comes up a lot. That phrase, wild type, like red eyes, certain wing shape, things of that sort. And then a mutant is the phenotype or allele resulting from a mutation. So you might see in your offspring, you'll have like 74 red and 24 white-eyed. And then there's one with brown eyes. That might be the result of some, random mutation. So we talk about forms of inheritance. If the presence of an allele in the genotype, that does not guarantee it will be expressed in the phenotype. If we're talking about, complete dominance. With complete dominance, this would be like your classic Punnett squares where when that dominant gene is present, then the dominant gene is expressed. The recessive allele is only expressed if you have both of them together. So for example, to have blue eyes, that organism would need a lowercase b, 2 recessive alleles inherited for that to be expressed. Whereas for the brown eyes, you only need one of those dominant alleles to be present for it to be expressed. Recessive alleles are alleles that are only expressed in the absence of a dominant allele. So that's what we see here. So another type of inheritance, so that what we just looked at was complete dominance. In this case, if we have codominance, what that means, drop my pencil, what that means is both traits will fully be expressed. So you might have a pink flower and a white flower, and you'll get a pink flower with white spots. So both pigments are fully expressed rather than blended. Incomplete dominance is when they blend together. So you might have red and white, and when they have both of those alleles present, they sort of blend together and create this pink color. So that would be incomplete. So some questions here to quiz you on that. What is the form of inheritance if you have a chestnut brown horse and a white horse? Well, you get something sort of in between. So what would that be? Incomplete inheritance or incomplete dominance. Okay, what is this form of inheritance? You've got a red flower and a white flower, and you end up with all red flowers. It's going to be complete dominance. And then this form of inheritance, you've got a white chicken and a black chicken, and then they get this black chicken with white stripes. So they're both fully expressed, so that's going to be codominance. Okay, so we can either do, just to run some of these predictions, you could do something like this. This is just your classic Punnett square. We call that a one-factor cross. You're only looking at one trait. Maybe this is eye color, height, wing shape, could be anything. But you'll notice we're only doing one trait, so one set of letters. A two-factor across is when you look at two. So maybe, round versus wrinkled seeds, and then green versus yellow seeds. So you've got two different traits you're looking at. That organism is going to have two alleles for each trait. So we have to map all of that out. These Punnett squares are typically going to be larger. And I'll show you a little trick so that you don't always have to make these massive Punnett squares. It makes it a little bit easier. Okay, so we also have some names for the generations. So the first generation that you cross, we call that the parent generation. These are the parents. So you might cross them with a Punnett square to see what the offspring could be. So what's in the inside of your first Punnett square, that's the F1 generation. It's the first generation of offspring. Then you might take that generation and cross them. And then the result of that would be the F2 generation, or the second generation of offspring. When we do a Punnett square, we're actually modeling two of Mendel's laws. And the first one is called the principle of segregation. So the two alleles of a trait separate from each other during the formation of gametes. So like what you put on the top and the sides of a Punnett square actually represent sperm and egg cells. So these are egg cells on the top, and these could be sperm cells on the side. So when a gamete is formed at the end of meiosis, we know that we end up with half the amount of genetic information. So when you make these sperm and egg cells to do a Punnett square, you have to split them apart. That's why you only put one of each type of letter at the end of each row or at the top of each column. So you're actually modeling Mendel's law that these traits, these alleles, will separate from one another. And then the other law that we model is this principle of independent assortment, that these genes located on different chromosomes are inherited independently of one another. So this gene in this sperm cell, this allele, is going to independently assort, and it's not going to have to be transmitted alongside of this other gene. So the alleles separate and independently assort to form these possible offspring. So when you look at a set of alleles, you should be able to tell whether or not it is a genotype of a parent or if it is the genes that are in the gametes. The way you can tell is like every person or every organism that is their normal body tissues, their genotype is going to have two of each allele for each trait. So that's how you can tell if it's a parent genotype or a gamete. So this one, since it has two of the letter P, that's going to be a parent genotype. Now this one has a T and a W, but doesn't have a set of each. So you know that's a gamete. This next one has two R's and two T's. So we know that's a parent genotype. When it forms gametes, it's going to split these. And maybe it could have a big R, big T, or a little r, big T. sperm cells or egg cells that it could pass on to its offspring. So what would be the possible gametes created from a parent with this genotype? So how do I figure out how many possibilities there are? Well, what you can do, and some textbooks will do it where you always have 4. But there's actually only, well, it depends on what the genotype is, how many options you're going to have. So the trick is to look at the parent genotype and look at how many options there are for each trait in that organism. So here we have a capital A or a lowercase a. So that's 2 different options. Here we have a capital B, a lowercase b. So that's also 2 different options. So to figure out what variety, how many different types of sperm and egg cells they can make, is we can actually just multiply those 2 numbers, and that's going to give us 4. So there are 4 possible gametes. Well, what are those 4 possible gametes? Well, we could have remember it's got to be split in half. One of each letter has to go into a sperm or egg cell. And we know we're going to make 4. Well, the first one can be capital A, capital B. That's one possible combination. Well, then you could also have a capital A with what? Lowercase b. Then we could have a lowercase a with a capital B. And then we could also have a lowercase a with a lowercase b. So those would be our 4 options there. So now we're just going to do a bunch of examples and cover a few different topics as pertain to Punnett squares. So the first one says, in pea plants, purple flower is dominant over white flower. A homozygous purple flowered plant is crossed with a white flowered plant. Well, if it's homozygous purple and purple is dominant, that parent's genotype is going to be capital, capital. It's crossed with a white flowered plant. Remember, white flower in this case is going to be recessive. So that parent's genotype is going to be lowercase, lowercase. Well, then we fill out the Punnett square and see what we get. So to do a Punnett square, we're going to put one parent genotype on the top, one parent genotype on the side, doesn't matter which way. And then we draw into the box from the top and the side of that box on the inside. So in this case, above is a capital F, to the side is a lowercase f. The convention is to always write the uppercase letter first. It keeps things consistent and makes it less confusing when you look at it in the end. So in this one, we're going to go above and to the side, so capital F, lowercase f. This one above and from beside, above and from beside. And what do you know? They're all the same. So the genotypic ratio is how many of these have different genotypes and what's the ratio between them. Well, they're all the same genotype, so the genotypic percentage, or ratio, will be 100% heterozygous, or you could say, it's a 1-to-0 ratio. It just is, that's all it is. Phenotypic percentage ratio is what it looks like. Well, they're going to be 100% purple. In pea plants, green pod color is dominant over yellow. Across 2 pea plants that are heterozygous. So they're both heterozygous. And then we got to put those genotypes above and beside the Punnett square. Then we fill it in. So the genotypic ratio in this case is 1, 2 of those, and then 1 of these. So my genotypic ratio is going to be 1, 2 to 1. And then I would specify what I'm talking about in that ratio. Phenotypic ratio, well, we're talking about pea plants, green is dominant to yellow. So 3 of the 4 are going to be green, right? Because they all have at least one capital G, and we're modeling complete dominance. So the phenotypic ratio is going to be 3 to 1, and that's going to be green to yellow. In pea plants, round seeds is dominant to wrinkled seeds. Cross a heterozygous round seeded plant with a homozygous round seeded plant. What are the genotypic and phenotypic percentages and ratios? All right, so genotypes. The first one is heterozygous round seeds, so they're going to be heterozygous with a homozygous round seed, so homozygous recessive. Oh, I'm sorry. Homozygous round, that's the dominant trait, so they're going to be capital R, capital R. So then we fill it in, fill in the boxes. All right. So in this case, we've got these two are the same, and these two are the same. So our genotypic ratio is 1:1, homozygous dominant to heterozygous. So that's going to be 50-50. Phenotypic ratio, they're going to be 100% round. Then we can do two-factor crosses. Sometimes these are really easy. Sometimes these, they're not hard, but they just take a lot longer. What you need to figure out with these is to figure out how many possible gametes can each parent make. And that's how big the cross is going to be. And I'll show you what I mean. So here it says that MP plants, tall is dominant over short, and green is dominant over yellow. We got T and G to represent these traits. It says cross a homozygous tall, heterozygous green pod plant. So homozygous tall, so they have capital T, and heterozygous for green, so capital G, lowercase g. That is crossed with a heterozygous tall, homozygous yellow, and yellow is recessive, so it's lowercase g. And then they give us this large 4x4 Punnett square to work with. Sometimes you will need one that large. Sometimes you don't. Depends on how many possible gametes each parent can make. So with this first parent, we got capital heterozygous. Well, he or she can only donate this capital T. There's no other option because there is no lowercase t. So one of their gametes will be capital T, capital G. The other gamete that this parent can make would be capital T, lowercase g. And that's it. So that is their possible gametes. The other parent is heterozygous and then homozygous recessive. So one possibility for them could be capital T, lowercase g. The other possibility is lowercase t, lowercase g. And that's it. So you take the gamete possibilities and put them along the top and along the side when you're doing these two-factor crosses. So for the top, we'll put the first parent. They could give a capital T, capital G. Second one could be a capital T, lowercase g. Then down the side, we have capital T, lowercase g, lowercase. And then we have to take all the letters into the box. So now we're going to have 4 letters on the inside instead of 2 since we're doing 2 traits. Because on the inside, we're predicting the genotype of the offspring. So that needs to be diploid again. So we fill it in, capital lowercase. Again, we're taking from the top and the side. Second one's going to be capital, lowercase. Then we come down here, capital, lowercase. And then we continue until that's what we get. Then we can look in and see what these phenotypic ratios are going to be. Now, genotypic, all 4 of them are different. So you actually have 4 different genotypes. So that ratio would be 1:1 to 1:1, or 25% each. Phenotypic ratio, you have, 2 that have at least a capital of each. So those 2, since capital is dominant over since tall is dominant to green, both of those are going to be tall and green. So you're going to have 50% tall and green. The other 50% are still going to be tall because they have a capital T, but they're homozygous recessive for trait G, which is going to represent yellow. So the other 50% will be tall and yellow. I didn't realize I gave myself a larger Punnett square for that. There's some of the answers. See, so this is what the textbook will do. It'll have, like here, gamete possibilities. These are not 2 different gamete possibilities, and neither are these. You could do a massive Punnett square like this, and you would come to the same ratios, but it's just a lot more work, and there's no reason to do that. So unless you have to do a larger one, I just wouldn't even do it. Okay, so we got all these other forms of inheritance, multiple alleles, incomplete dominance, codominance, blood typing, sex chromosomes. We'll have sex-linked recessive and that sort of thing. So since alleles in these forms of inheritance are not dominant or recessive, only capital letters are used, and the alleles are distinguished using superscripts. So we'll look at some examples of these, how we do those in Punnett squares. This is good when you have like those flowers where you have a red and a white, and then they cross and make a pink flower. Because both traits are going to be present and expressed in some way, we make them all capital, and then we use superscripts to denote the different varieties. Because maybe you have red, white, pink, gray, all these different colors, but they're all going to have an influence on the offspring. So we have to represent them capital letter and then a superscript to change what they do. And that's what this is showing you here with this snapdragon. All right, so let's try one of these. It says cross a red flowered snapdragon with a white flowered snapdragon. R1 is for red flowered, R2 is for white. So the parent genotypes are going to be R1, R1. The white flower is going to be R2, R2. And then we'll fill this out. Really, we'd put a 1 up in the superscript there. R1, R1, R2, R2. Then we fill it out. And they're all going to be like this, which means they're all going to look a mixture of between the two because we had red and white parent. So now 100% of the offspring are going to be pink because this is incomplete dominance. They told us what kind of dominance it was. So the genotype is going to be 100% R1, R2, and the phenotypes are going to be 100% pink. All right, here's another one. Cross a pink flowered snapdragon with a pink flowered snapdragon. Well, the pink is going to be that blended, so their genotypes are R1, R2. And then we're going to cross them. Then we fill it out. So a little bit different here. So now the genotypes, let's see, we have one like this, two like this, and then one like that. So our genotypic offspring percentages would be 25% R1, R1, 50% R1, R2, and 25% R2, R2. Phenotypic offspring percentages are going to be the same because you're going to have 25% that are white, 50% that are pink, and then 25% that are red. Now, multiple alleles is when you have something like this with blood types. So blood type, if you have the gene for, surface red blood cell surface protein A, if that gene is present, then it's expressed, and you have this little surface protein that is on the surface of your red blood cells. And if you have the gene for blood type B, then you're going to have a surface protein that looks a little bit different, and they use the letter B to describe it. If you're AB, you'll notice you have both. You have this little triangle shape, and what they're using here, triangles and circles, to represent that. If you're blood type O, then you don't have anything on the surface. Now, this is ignoring what's called RH factor. So these could all be A positive, negative, B positive, B negative. So we're ignoring that. But for now, we'll just, keep it a little bit more simple. So that's what actually is your blood type. So like if you're blood type A, you can't receive a blood type B transfusion because your body's not going to recognize blood type B cell surface proteins. Those antigens are going to trigger an immune response. So it could be deadly to get too much blood transfused from someone's blood that's a different blood type because there's going to be a massive immune reaction to that. So when we talk about who can receive blood types from who, see, do I have that on here? I do. Then that's going to be what we have right here. So like if you're blood type A, they use I and then A or IB, IO to denote all these different blood types. So if you're blood type A, you could be IAIA or IAIO. Because IO doesn't code for any surface proteins, you're still blood type A, even if you're heterozygous in that sense. Same for blood type B. So if you look at what they can receive blood from, you can only receive blood from people whose blood type is either the same, or if it's different, it's okay as long as that difference is blood type O. So for example, blood type A could receive a blood type A transfusion, or they could receive blood type O. Either way. Since blood type O doesn't have any surface proteins on it, like we saw here, that's not going to cause an immune reaction. Same for blood type B. And then blood type AB, since their body's already used to recognizing blood type A and blood type B cell surface proteins in their blood, well, then they can receive blood type A, blood type B. They can still receive blood type O because that doesn't have any surface proteins. It's not going to trigger an immune reaction. And they can even take blood type AB because their body's already used to both of those cell surface proteins. And then blood type O can only receive type O because their immune system is not used to A or B. So they can't have A, B, or AB. So all they can receive is type O. So this makes type O a universal donor, and type AB you could think of as a universal recipient because they can receive any blood type. If a father is homozygous for type A and the mother is heterozygous for type B, what are the possible blood types of the offspring? Okay, well, homozygous for type A, heterozygous for type B, and then we'll fill that out. So one application here, well, we'll look at the options first. So one option is they could have a child with type AB blood or type A blood. These two parents could not have a child that is blood type B. So sometimes if there's like the accusation that someone is the father of a child and maybe they're claiming that they're not, they based on their blood test results, they could tell if it's even a possibility that that child could have the blood type that they have based on the parent's blood type. So it could be a way to check for that, to see if it's an option. If a father has blood type AB and a mother has type O, it's the only way to be type O, what is the possibility of them having a child with type O blood? So then we fill it out. Looks like it is 0 because none of those are IO-IO. Okay, so then we also have genes that can be pinned to sex chromosomes. So in humans, that 23rd pair is where we're going to see the sex-determining chromosomes that determine the sex characteristics and traits and development of humans. There are abnormalities. So generally, XX is going to be a female. XY will be a male, unless there's some kind of strange, usually often rare genetic abnormality. And they'll probably still develop into a male or a female. There might just be some symptoms or side effects. So, for example, males can be XY. They can also be XXY, which is Klinefelter syndrome. And that extra X chromosome creates a lot of health issues. So, any abnormality from those 23 pairs with the 23rd being the XY or XX is going to create some issues. Or they may even have the normal chromosomes, but there's some kind of mutation or disease that's been inherited on those chromosomes that can affect potentially how their genitalia develop or their hormone levels as they grow and develop and go through puberty, or even how they react to testosterone and estrogen could be affected. So anyway, the first 22 pairs we call autosomes, and the last pair is the sex chromosomes or sex-determining chromosomes. What is the possibility that a man and a woman will have a son? Well, a man is going to be XY, a woman's going to be XX. So what is the probability that they're going to have a son? Well, we could fill it out. Well, XY is a male, so that's going to be 50% chance for a son, 50% chance for a daughter. And if you look around the world, it's about 50-50. And this is why a male is going to donate either an X or a Y in his sperm cells. A female can only donate an X chromosome in their egg cells. So it's basically a 50-50 shot. And that brings us to our second lesson.